about structure determination and secondary structure, and a little bit back to the issue of comparative modeling. There is the next lecture that will not happen is the one on May 29 because of Ascension Day, so that's next Thursday. Recap on 3D prediction by comparative modeling. So again, you can compare sequences. So dynamic programming, in fact, again, does not compare structures, it compares sequences. This graph is valid for comparing sequences. So Lothar suggested to call it daylight zone, I like that term. Called it safe zone so far. Now here it's as daylight zone, so where just by comparison of the two sequences you can infer that the two have a similar structure. Twilight zone, you no longer have a clear signal. Midnight zone, you don't see it at all. Here, and that's another kind of thing that would be asked in a talk, what's the difference between sequence, profile comparisons? Where can they enter in terms of this idea? And simply the idea of comparative modeling is that proteins with different sequences that are evolved from a common ancestor most likely have similar structures. So if I know the structure experimentally for one of those, I can predict this structure for the others. By generating the alignment, and then for every single position, look up, say the purple would be the one for which I have the structure, then look up for every single residue we have in alignment, the corresponding position in the purple, and just in that particular case, I would say that in fact green is exactly equal to purple. You see that purple here deviates from what is known to be the green structure. That would be a mistake of the comparative model, but overall you see that the purple and green is relatively similar, so overall this model, comparatively built model, would be relatively accurate. The two main methods are modeler and Swiss model, and comparative modeling is applicable to about 1/3 of all the proteins. Again, we currently know about 55 million protein sequences, and for only about 100,000 we have the three-dimensional structure. And the gap in between these two extreme numbers, so 1/3 corresponds to roughly 18 million, and so between 100,000 and 18 million there's a lot to do, and that is exactly the ground that is tread by comparative modeling. Now let's get back to comparative modeling again in terms of assessment. So again, in principle, the idea is, or the reality is, and that is a paper from Anfinsen, published in Epstein and Anfinsen, published in 1961. They said that if you take a stretched out sequence, amino acids, so say you take 100 residue protein, you throw it into a solvent, water, it adopts a unique three-dimensional structure. Okay? And this unique three-dimensional structure is necessary because structure relates to function. If it would slightly change sometimes, because today is Tuesday, it would have a different structure on Friday or something like that, then it would have a different function, and that would be largely not good for your body. So the fact that it always adopts the same three-dimensional structure is very important. Now if that experiment is true, and for many proteins you can make that experiment. Today we no longer know whether that is true really for all proteins if we find the right conditions. For most proteins, we today know that for many proteins this is most likely not true. But for some, clearly it is true. So for those for which it's not true, we assume that in the body there is a sort of safe area where things are broken up, let's simplify that, into small parts where that again becomes true. Now back to that statement. If the protein thrown into a solvent adopts a unique three-dimensional structure, then the problem of going from the sequence to the structure, in principle could be done by physics, or chemistry, or biochemistry. Because in principle then, it is something that has to do with the features of the chain and the features of the solvent, right? So that means we could just simulate it. Right? What do you believe is the problem? Let's get back into some other issue here. That's the statement I made. In principle, the folding of a protein should be predictable from first principles, from physics, from simulations. And indeed, you will find that the problem has been solved. It has been solved for the first time in the 60s. There was a big article in the Washington Post has been solved again in the 70s. There was a New York Times this time, and in the 90s, another Washington Post article. And as you can see already from this, if it's solved 3 times and 3 times it makes head news, then something is wrong, right? You cannot solve the same problem in the 60s and then have a New York Times article about just having done it in the 70s. And that brings us to the issue that in fact it has not been solved. So the problem is not so far solved. What do you believe is the problem in the problem? So again, I have all the amino acids. Say I take 100 residues. So I have 100 different amino acids. I have solvent, this water. I can just simulate the physical movement of this chain. What's the problem?
Maybe it's too complicated, so you can't simulate.
Why? I can simulate the moon and the sun. This is much smaller.
Yeah, maybe there are quantum effects.
So the suspicion is there are quantum effects, and that certainly is true. That certainly is a problem. And in fact, that relates to, in many ways, to the Nobel Prize in computational biology last year, or the 3 Nobel Prizes. You're right. But assume that would not be the case. Assume there wouldn't be quantum mechanical effects that are not easy to treat. Assume this could just be done by classical physics. Yeah?
Oh, if you predict the movement of the sun, or if you predict the movement of astronomical bodies, that's actually not possible to do analytically, because you have the 3-body problem, and it just, so what you can do is you can put a lot of computing power on it, and then it works out to that you only get very small errors. But in modeling the 3D structure, or predicting the 3D structure of a protein, you have a much bigger problem, or the problem size is much bigger, and it's probably an NP-hard problem to match the subsequences of a protein together for how they fold, because you just have a multi-dimensional space of how the folding can happen.
So let me just summarize for those in the back who may not have heard, although he spoke, thank you, spoke very loud. So statement number 1, sun and moon is true, we can do that. I'm misleading you because sun and moon is 2. And he pointed out correctly that already at the number 3, sun, moon and earth, the problem gets very complicated. In fact, it gets so complicated that there's no longer a trivial analytical solution. You can do that by throwing a lot of CPU onto it. Now in this particular case here, for a 100 residue protein, each residue has about 10 atoms. So we would talk about a 1,000 particle problem. And that is highly non-trivial. So you can throw a lot of CPU at it and still not solve it. And now we get into a couple of issues. One issue, and at some point I'll get back to that. One issue is called Anton. Anton is a very large computer that stands in New York. Is done through the initiative of David Shaw. Actually, David Shaw is heading the institute or financing the institute that built Anton. Anton simply is a special purpose machine. So with a lot of money, really a lot of money, they have been building for more than 5 years a machine that just does fold proteins and just tries to crank the numbers and address all the problems that are related to this issue here of predicting the structure of a protein in a way that is optimal for that hardware. So they changed the hardware, they changed the software, they did everything so that the whole thing matched 100 people 5 years or something like in that ballpark. You get an idea about the magnitude of the enterprise. It's a unique thing. And Anton now can, so they have published 3 years ago, I believe, the folding of a short peptide, propeptide, of about 30 residues. That's as far as we get. We can get a tiny fraction of a second for the structure formation of this one, and that is the breakthrough of the century in many ways. That's by far the furthest you can get. IBM threw something at it a decade ago that is many orders of magnitude away from what this machine can do. But again, so even with more computer power than we actually have, so purpose building a machine for that, for many years with a lot of money, we still don't get at folding one single protein of 100 residues. But now, what is the problem? So I said there were 3 articles that announced that the problem had been solved. Why is it so difficult to assess whether we can do that or not? What do you believe? So again, you know, I don't know, it's not easy for a scientist to make headline in New York Times or Washington Post. So it's a major event. It's not like some journalist has heard something may happen and jumps on it. They're very careful people.
It could be easy to have a result, but maybe very difficult to show that the model that we compute are wrong. So maybe in 3 years, one scientist say, hey, look, I've got a great result with this algorithm or with these methods, but they need 10 or 20 years to just destroy the arguments and say this was wrong.
So this is interesting what you say. I'm not sure whether everybody, let me just repeat it. The way I understand that statement, essentially, it is complicated to verify, so there's a computation done, and it's very complicated to verify that computation, and it may take 10 or 20 years before we find out that's wrong. I believe that your rationale comes from mathematics. So you may all be aware, you submit a paper in mathematics that is ground shaking, and it's not unusual that you wait 3 years before it's reviewed, 5 years before it's published, and maybe a decade before the experts in the field really know whether it's true or not. Okay? I assume that something like that is what you have in mind. But in this particular field, so let's assume we build a computer, or we have some theoretical mechanism by which we predict the structure, all right? And in fact, these are more conceptual solutions than big computer solutions. Those 3 instances here. Now, why not just take a structure and look whether it's right or wrong? Let me take him because he...
Because the knowledge of all the structures in the 60s wasn't that great and wasn't that big, and then maybe during the 60s they had models that could explain everything that they knew, and in the 70s we had new knowledge and say, oh wait, with this new...
That's a great idea. So for the people who haven't seen that, the argument is, well, in the 60s we didn't have that many. Maybe somehow we could do something for the ones we knew. But then we got new ones and they look very different. And there's a lot of truth to that. I'll get back to one aspect of that truth in a minute. But that brings me to this question here. Now we have the 2010s or something, 2014. Now somebody publishes a method. How can we check it? Wait 10 years?
Well, it comes down to a very basic fitting problem, which or if it's wrong, you get an overfitting problem, where you can only
How can you avoid that?
compare your results to results that you already have, and then you have to develop your method looking at one set of proteins, and when you're done developing it, you have to reserve some other set of proteins that you can use to verify it. But both of those sets are already known sets, and then maybe it will only be optimized for that set of proteins that we currently know.
So the statement here is essentially leading to cross-validation, stating that you have a set known of proteins. The proper procedure would be to split that set into 2 sets or various sets. Let's not discuss the detail. We will discuss the detail soon, but different environment. So we have different sets, and we take one set to optimize our method, and the other set that is completely independent of the first set to check whether the method works. That seems to be a very clear procedure. Is that enough? And actually, so that procedure is not an invention of 2014. In principle, that procedure was around the 90s. Could have done that then. Any idea how we could do it? Any suggestion how to find a way to avoid that without waiting 10 years? Waiting a little bit is okay, but 10 years is a long time. And certainly in biology. So in biology, again, this is a field of high competition in terms of speed. You have to be the first to publish something. You publish a new fact as the second. You do not make the top job. And the citations do not go to you.
Maybe we could find a way to somehow take into account new protein coming in the way that we are not taking only the protein, but just saying I'm considering possible models of protein that are likely to exist, but we don't know yet that if they exist or not.
That's an interesting idea. So the idea essentially, in fact, that has been tried, and there's a longer story to that. So the idea is, let's just have a couple of methods that predict a bunch of models, put these models on a stack, and wait around until somebody has some experiment that says one yes to one or no to the other. So it distinguishes between the models. That is a great idea. Here's a similar solution called CASP, Critical Assessment for Structure Prediction. And the idea there is you look at a bunch of proteins, or you ask all your colleagues who are experimentalists, is there any protein that you're going to release, for which you're going to release the structure, the experimental solution, over the next half year? If so, at this point, nobody in the world knows the structure. But if so, are you willing to in fact submit the sequence to some server? So now the sequence sits on that server, and people who develop methods can try to predict the structure for that sequence. At this very point in time, nobody in the world knows the structure, including the experimentalists who in fact do the structure. And then at some point when this manuscript is, or when the experimentalists are at the point where they write up the manuscript, the prediction season closes for that target. So meaning that everybody has to send in their model, and after that point, typically defined as somebody could have seen the structure outside of that lab. So after that point, nobody can predict anymore. We collect for several months, so that is referred to here as the collection season, April to May. April to May we set up the experiment. June to August is the prediction season. And then comes the point when all the targets have been submitted to. September, November is the period when the people who in fact look at the quality of these models. They discuss that for 3 months, and then everybody meets at some point in December. Right? This is the CASP experiment. The CASP experiment has been running every 2 years since 1994. So it's in its 20th year, and what this one here simply shows the number of groups that have been participating in CASP. So this is strongly growing. So what CASP shows, yes, sorry.
But with this kind of thinking, we will have a kind of overfitted model, because we are just targeting, we are giving some proteins a very particular role in the development of the model. So we know that our model will be tested on this kind of proteins, so we do everything that for our model to be good on this kind of indicator, but it's not that very honest.
So now, in fact, I don't know how you sort of suspect that. You put your finger on something that is a real problem, and that I would prefer to hide for today. You go into the detail. Namely, the issue that the moment CASP is defined the way I define it, everybody knows what the criteria will be to predict. And they can over-optimize their models to predict, is the statement. And again, there's some truth to that. Now, the other part of the statement is the kind of targets that we send are predictable. Well, that's not quite true. So the kind of targets we have here are protein structures that are today not known. Protein structures that some experimentalist feels are valid targets to work on. And if I in fact can predict how they looked, you may call that overfitting from today's structures. But if I can predict what happens over the next 3 months in terms of experiments, that may be overfitting to what we do now, but is a great achievement in principle. So now your argument must then be, in order to be consistent on that part, again there's a detail that I completely give you, that's a strong one and I don't want to discuss it. But now the strong attack argument must be that the next structure out there must look very different from what we have today. And very different in terms of, I cannot learn anything from today's data. on those. But that actually is an extremely strong step. Because now getting back to the Anfinsen-Epstein story, if the protein, or many proteins at least, put into solvent know how to do it, if Anton can do anything, then there is some common principle. And no matter what the proteins will look like in 10 years, they will still obey that common principle. Again, you may call that overfitting, or you may call that fitting. True, it's fitting to data that we have, but we believe that today's structures are some frozen reality of what is possible. We assume there will be things that we cannot do today. And there, again, let me come to that in a minute again. There are principal obstacles to the way we do structure determination. Some proteins we just cannot get, or are very, very difficult to get. So we assume that in 20 years there will be things that are very different from what we have today. But if we look compared by many ways what we have today and what we had 20 years ago, then again there's an overstatement in this here, but in some sense they are somehow similar. Which may be because the ways in which we determine structures are still similar to what they were 20 years ago. And that may be the circle there. But again, so in principle, that should work. Now the criticism says, the problem is it may overfit the data. But actually what turns out is that the only thing we can really do very well is comparative modeling. So if this overfitting aspect would be a dominant one, then many things should look good. And actually CASP, if anything else, reveals that largely structure prediction fails. That's what I presented to you in the last lecture, comparative modeling. So copying the coordinates from another protein for which I have the experimental structure that has a similar sequence is the only way that really is secure. And that gives us good models. We cannot predict 3D structure from sequence yet. Again, Anton can do it for small fragments. That's as far as we got. And there's a lot of development that has been done over the years, and you see in the number of groups that participate, there's a tremendous effort in this field. There are many, many improvements over since 94 to this one here shows 2004. Yeah?
So in this CASP, you get some, let's say, approximate answers, for instance, using this comparative modeling, and then you compare it with some perfect answer, yes?
Yes.
But how do you achieve this perfect answer?
So again, the idea of CASP is, so the question was, I have some approximate answers, so the comparative models, and I compare them to some true answers, the experimental structure. Where do I get them from is the question. And again, the idea in CASP is we send in the sequences and the models while the experimental structure is not known. But some of the organizers of CASP know that the structure will be there, yeah, hopefully.
How will there be?
There's an experimentalist working on it and determining the experimental structure. And in fact, for some of the CASP's targets, so a typical CASP, we get back to that number, would have 100 predictions done. And maybe only for 70 of those, the structures really will be there in time, will be assessed. For 30, the experimentalists said we will finish in June, but they will not finish in that time because there were problems that they didn't anticipate. But for the 70 for which you have a structure, you have a new experimental structure, again, that may be similar in terms of the technology of determining the structure, but it's a new structure that nobody at this point of submitting the model, the prediction, has seen.
Yeah, but this new structure is found out not by computing, right?
No, experimental. So this new structure is an experimental structure.
Do you have some degree of reference that you can deviate from?
You mean for the experimental structure or the model?
Like using the CASP, and do they have any specific reference that we have to follow, or we just randomly let go through it?
So again, there is one truth for every one of these 70 that come through. There's one true answer. And that true answer is there's no error allowed. That's the true answer. And then the models are assessed with respect to that true answer. And the way it happens is that again, I showed you the numbers here. There are thousands of models in there. For every single of these 70, you have thousands of hundreds or so of models. And then you compare what is the best and what is the worst. And then you hope that there's one method that is always better for all the proteins. And to some extent, that's true. Not always. Now, again, there are many, many, many improvements, and the major improvement, in fact, is the degree to which we go further down into the twilight zone. So the degree to which we apply the first step, comparative modeling, can now be applied to distances of sequences that it couldn't 20 years ago. That's the major improvement. So we can map, and we have more structures and more sequences. So we can map more of the things that come in new to things we already know. That's a breakthrough. Now, when the first CASP happened in '94, many people, in fact, had assumed that their methods would work much better than they actually did. So this CASP meeting was quite exciting because it was a big surprise for everybody in the room, or for many, many, many people in the room. At the first CASP, almost everybody had come with their own programs. Yes, you know, some aspects of these programs that I talk about today, for instance, secondary structure prediction, have been done by other groups, but you know, why shouldn't I do it in my group? I trust myself more than my colleague. What happened after the first CASP is there were winners and losers. And in the second CASP, immediately everybody used the winner method to improve their own method. That got into a world of meta service. So service of service is service, and completely brought a new reality into CASP. A little bit too much maybe. What these lines show is somehow the improvement of a CASP. This is the target difficulty, that somehow the success up here is completely right. Sorry, down here is completely right. No, up here is completely right. Down here is wrong. So difficult targets are less well done. Easier targets are better done. And there's some tendency that for later CASP, this curve goes up. So there is some tendency to argue that the field has improved. Problems with CASP are, the first thing is the limited number of proteins. There's a lot of effort to find the experimentalists who submit their proteins. Typically it's 100 proteins. That's one issue. Now we get into another issue here. The issue is, for instance, assume that method A uses 53 proteins and reaches performance X. Method B uses 35 and reaches X plus Y. Is B better? Assuming Y is a positive number.
I think that we cannot say that A is better or...
What do you need to know from me?
No. Because you have a limited window, you are much prone to errors, and if you have a wider range, then you are more specific and you will have more precise and specific.
Yes, your suspicions are both right. But I said why. You never asked me what I mean by why. You said no before you ever asked what Y means.
And we have to have something to compare it to, the result.
Yeah, so that is basically the statement again. We have a true structure, and both are compared to this true structure, and in comparison to the true structure, it reaches X, method A. And comparison to the true structure, method B reaches X plus Y, so a little bit higher. But the question of, obviously here the word better means, can I really get away and can say that method B does something that is significantly different, significantly better? And there are two comments that were negative, and I'll get back to these. So the first question, obviously, are 100 proteins enough, right? And that brings us back again, so what is the diversity, to which extent do I get it right? The other question is the why. So how big is the difference? Is that significant? How would you look at these two problems? How would you answer those? So again, you both gave emotional answers, I would say. You said essentially something is fishy with what I'm proposing to you, and anyway I'm asking you a question, and you're right. But how would you define scientifically how to do it?
So earlier you presented a graph which denoted the difficulty of protein and the performance of the CASP predictions, and then with that we can maybe compare that So the protein with 53 is this difficulty, and the average is maybe this much, and the 35 is slightly less difficult, and then we can compare according to the graph if y is enough.
Exactly. So just look at the problem in terms of solutions. Say one method had a distribution like this, and the other one had a distribution like this. Then clearly you would see that these distributions overlap largely. So the average simply is the statement is not enough in order to make that decision. How big y has to do with the width and the difference, right? Here of the two. So you need that information. But there's something else that I was sort of hiding under the rug. I said method A has 53 cases, method B has 35. Is that okay?
By cases, you mean that they use B, 35 proteins to be built?
Yeah. No, not to be built, sorry. So again, CASP, in this assumption, CASP has submitted 100 proteins, or you submitted 100 models for 100 proteins, or some subset. Maybe you couldn't do every single one, because your machine crashed on some of them. So, and for some of them you didn't get the true answer. End of the story, the ones that the assessors could assess for your method were 53. That may be because you only submitted 53, or maybe only for 53 of the ones you submitted, they actually had structures, okay? The other method, you submitted only 35. That's all, yeah?
Yeah, maybe the other one just took out the balances, so it's...
Yes, so we clearly have to, oops, wrong direction. We clearly have to compare apples with apples, meaning that we have to first ascertain that we are actually talking about the same proteins here, right? 53, 35. 35 is a smaller number, but they may, as you suspect, be one group may have just chosen the easy ones, right? So now, let me phrase the question slightly different. So, I could look at identical subsets, right? In this particular case here, I assume that the number of identical proteins in the identical subset is 11. But I want to compare, in this particular case, I can't remember, 7 or 8 methods. And these 7 or 8 methods have all submitted 29 sequences. So I could look at 29, 7 methods using 29 different ones, or 11 identical ones. What is better? Just it's a wild guess. Your smile is totally right. You cannot know.
I would say that everything should be...
Yes, and so that's what the CASP organizers have always argued, that we should use as much information as we can. 11 is just a tiny number. Again, we started in this particular experiment, I can't remember whether it's quite true, but we may have started with a submission of 100 models. So boiling it down to 11 of 100, just because we couldn't agree between 7 different methods, sounds like a very scary, extreme jump. 29 at least is 1/3 of all the initial ones. That sounds better, right? So I'm going to present again, and this is not type of the complexity that I asked for in the exam. I would hope that you have an idea of dynamic programming in BLAS, but I really don't expect you to understand the complexity. There's some level of accuracy here. These are the number of random draws. So essentially the way I answer that question that I pose here, I have 7 different methods. For the 7 different methods, I have a much larger set and therefore the true answer. Now I sort of simulate one particular CASP. So I compare 29 versus 29 different ones against 11 identical ones for each method, and then I repeat. I just take a random draw of my large set. I repeat and I repeat and I repeat, and I ask, how is the ranking changing? And you essentially see, or you don't see because there's too much data, but if I gave you enough time, what you would see is that 11 identical is in way better. Although the number 11 is very small, way too small in fact, as we can see, using 29 different ones is much more dangerous, because the probability that one of them will take the easy ones away, if the sample set to pull this from is large enough, is extremely high. So here the outcome result said that for the 29, every method in one of the pseudo casps here, this is sort of 30 times casp simulated in the computer, in one of these pseudo casps, one of them was always the winner, although in the set of 7 there were clearly losers and winners. But doing it this way, you could not distinguish. Doing it this way, you could also not quite distinguish between, because 11 is just too small a number, but 11 turned out to be better. So ultimately what you have to do is you have to really compare apples and apples, meaning you look at a method A and ask, is method B doing better? For this question here, you have to look at the statistical significance, so the deviation of distributions, and you have to use the same data set. So you have essentially a set of pair comparisons. And for every pair comparison you get as much, bless you, as many as possible. So this could be built on 28, this may be built on 13, and that may be built on 15 proteins that I have in common between these two. So every entry here in the matrix now is built upon a different sort of background distribution. But since my value is statistical significance, somehow looking at the distribution should handle much of that difference. And then ultimately the question becomes, the winner is the one that clearly always stands out, if there is such a case, yeah?
With this kind of situation we can have a circle.
So there are clearly examples. You can easily imagine that in this particular kind of situation you have a circle. So meaning that you can in fact not distinguish between method A and B. Would that be a problem?
I think that we have A is preferred to B, which is preferred to C, which is preferred to D, which is preferred to A. So
no, that.
We have a non-positive.
So if it is, oh sorry, I misunderstood what you said. So a cycle is unlikely if A is significantly better than B,
yes,
and B is significantly better than C,
yes,
then it's very unlikely, and C is significantly better than D, then it's very unlikely that D is better than A.
Yes, but as soon as we don't take identical proteins for compare A to B, B to C.
Oh yes, you're right.
We can imagine that.
Oh, that's a good point, and that's a frustrating point. Let me define frustrating. Thousands of people have worked on CASP. That was the pinnacle of what we have come up with. I have never thought about that problem. It's true. Hasn't happened. So why hasn't it happened? Because now we get back to it's aligned with your previous suspicion. If we pushed or pooled the data that I used, so the experimental structures, from one particular corner in space, then that in fact can really happen. Example, you could imagine that the first 3 predict only alpha proteins. And what I compare here is beta proteins. And then I make the mistake because I'm completely comparing different things. That may be a problem. Actually, I haven't seen that problem happening. But that is in principle, this is a problem, yeah? I take that argument. So that problem relates to, becomes higher the more the common subset is a tiny fraction of the space of all the ones that we had. The closer the subset that I compare any pair to the full number, the less likely this problem will hit. Meanwhile we have, so I showed you 11 of 100 before, meanwhile we have come to situations where many models that are compared hit much larger fractions. But that somehow is related to a problem that really is there. But again, so the conclusion from comparative modeling is that comparative modeling works, is fairly accurate, and is in fact the only accurate way to predict protein structure. Is as good as the complete template. So I can in fact not do much better. So what I get, what I read off the coordinates of the other protein, the experimental structure, is as good as I get. I can typically, in principle, I could use clever programming to improve over that, because my sequence is different, and sometimes I can. But I cannot do that consistently, not always. Not all methods always get the improvement right. It's mostly driven by the accuracy of the alignment. So that's why the dynamic programming is so important here. The first mistakes you make will never be corrected. Loop modeling, so things that flop around that are not very constrained, is a problem. Now, let's get to the issue of how do we determine experimental structures. Those are sort of the residue, the residue level, the trace chain seen here. Do you have any idea how you do structures experimentally?
Well, I think what you do is you chemically turn the protein into something that is amenable to be somehow scanned with imaging methods.
That's true.
Maybe you have to cut it into small pieces before you do that.
That would.
And then you use some very high resolution imaging methods to scan it, probably something like electron microscopy.
Let's get back to every single part. So the first statement is, I'm misusing what you're saying, okay? Just the one statement is I have to use imaging, advanced imaging technology. That certainly is true. But now the major issue here, electron microscopy was one issue. The atom is too dynamic and too small to just stare at it under the microscope. I will show you images of very, very advanced electron microscopes, what type of resolution, so what type of detail they actually can see. But another statement that your colleague made was that I have to break it into pieces. Now the problem is that you can hardly, so again, yes, in terms of domain, we want to look at an individual domain because of a variety of reasons, but the domain we believe we cannot break further down because we believe that when we break the domain down, we actually miss the important interactions. We miss the important picture. So breaking down we cannot do. And again, we cannot look at a microscope easily. And I don't have a date on here, but of the then, maybe that's half a year ago, 84,000 structures in the PDB, 75,000 have been determined by X-ray crystallography, 8,000 by NMR, and 400 by electron microscopy. And I'll get into these three methods a little bit, well, one method per slide, so it really is not, it's not a level of detail. So I have mentioned to you that the density in the cell is that of a solid state. In fact, there are proteins that are so densely packed that you can just scoop them out of the cell and you have a crystal. Okay? Once you have a crystal, you can just shoot X-ray beams at it. And by X-ray crystallography, when you go to the subway here, you see how crystallography works in principle. And that is essentially the same idea here. The way it shatters the light, reflects the light, it tells you how in this crystal, where in this crystal the atoms sit. It's an ordered crystal. And here we get to this first question that I answered this morning. So the crystal may contain pollutions. Those may be things that you see in the PDB structure that have nothing to do with the protein. They have nothing to do with its biological role, but are simply, let's call it noise, that comes about by the way the crystal has been formed. It could be environment. It could be in the solvent. It could be an additive that helps the crystallization procedure. And now we get into the next thing. The moment you don't, so most proteins are not at that level of density, although the cell has that level of density. You typically cannot just go there, scoop, take the crystal out. Typically what you do is you take the protein in vitro and try to aggregate it, so have a lot of it, to concentrate it. When you have enough of it, you hope that a crystal is growing. And that is an example of a crystal. When you attend talks from X-ray crystallographers, it always will show you one of these crystals because they are beautiful and because X-ray crystallographers, from the way these crystals look and diffract, they can already see something about the quality of what they're going to see next. So just looking at the crystal, don't have him on here, but Kenru is the kind of person who in fact would have, no, but certainly Wayne Hendrickson, this name is not on here, is sort of a modern crystallographer from Columbia University. He can look at these crystals and sort of predict how good that is. So the diffraction pattern, then you get an electron density map. Now here we are in this situation where we actually have some, let's call it model building. An electron density map is a probability distribution. And now you have to in fact trace the chain, the protein, into that electron density map. You see in electron density, you do not see residues. The protein does not have numbers, residue 1, 2, 3, 4, and then you look at the X-ray and you see these numbers. You have to, from that electron density map, by fitting, get out the atomic model. So which density, which signal, visual signal, corresponds to which residue? That's a not so trivial work to do. There are many programs that improve this, and today we're down to a situation where this is doable in just days. That's as fast as we can do that, largely due to advances that have been done by experimental biologists or by structural biologists, experimental structural biologists, to in fact do the structure determination and have developed a lot of these methods underneath, the computer programs and the math and everything behind it, the biochemistry behind it. And then you get to this level where you really have atomic models. The other way in which you can do it is by NMR, yeah?
Could you repeat how to get the crystal from the protein?
So the crystal, again, let's assume that we started from a gene. So all we know all human genes from sequencing. Determining your sequence is relatively easy. So for every single protein, I know the sequence. Now I can take that sequence and in fact build a protein. That is very expensive. So I cannot easily do that. Typically the way I do that is I take the DNA and I put it into some bacterium and force the bacterium to express that protein. Now that could be, I take E. coli and I want E. coli to express a human protein or an E. coli protein. Funny enough, in fact overall, so I'm part of a consortium, the NICOMS consortium, where we found out that if we use 10,000 proteins for this, then E. coli is not doing better on its own proteins than on human proteins. Or on proteins from other organisms, funny enough. Anyway, so I make E. coli express this, if E. coli is my thing. Express means it actually produces a protein. Now I try to have a lot of that protein. Microgram, milligram. Certainly a milligram would be great. When I have a lot, I put them on wells, so on dishes, on Petri dishes, on places, and then I try crystallization experiments. And now we again get to the pollution. Crystallization experiments, I throw things into there, additives, hoping that some of them, under some conditions, in some cases I have additives, in some cases I don't. And I have different conditions in terms of humidity, temperature, and so forth and so on. And I just try out as many conditions as possible, and I keep working on that. This is an art. Art is defined as every successful structure determination lab has their guru, who is the person who is really good at that and has been doing that for many years. And some people just manage and some don't. And that distinguishes to some extent between good publications and not. And a lot of that is not, if you want, science in the sense that you can really predict it. But still, the good ones can do it again and again. In that sense, the artist knows more about the problem than I. Okay? But in principle, that's the way you do it. You have enough of the material and you wait for a crystal to grow. Okay? Which again, may sound surprising that proteins grow crystals, but the way they operate in the cell is a very high concentration. So it's not that far away from reality. Okay, NMR. nuclear magnetic resonance, and again, in terms of numbers here, now this is about 1/10 of the ones that I have before. The great thing begins with this NMR tube, and that is really one in operation, meaning that you don't, in this particular case, you don't have to grow a crystal. Take the protein in its solvent, in this microtube here, and put it into an NMR machine. Now, here the problem for NMR is a little bit the size of the machine. So this is the New York Structural Biology Center, those are the NMR machines that then, there's a 900 megahertz machine. I thought there was a person, yes. There's a person up here, so it gives you somehow an idea about the scale. This building here, this room here is larger than this room. And one thing is clear, you enter that room with anything that has a credit card on it, and you're sure to not worry to use that credit card again. So these magnets have a strength that is incredible. Another way of explaining that is the mere weight of these guys. So Manhattan, as some of you may know, is built on very, very solid stone, sheetrock. And the sheetrock is not enough. It needs support in order to put a machine like that in there. It's just a mind-boggling scale here. So you begin with this tube, where you don't need to grow a crystal. You have a nice NMR tube in solution, much easier, but then you have to get it into that big machine. The next issue with NMR is, in fact, so you get nuclear resonance, and in this nuclear resonance you have more of a problem than in the electron density map to trace your chain, to find out which residue do you see at some point. And therefore you get more than one model out of it. That is sort of indicated here by these different green lines. So each one of them, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, like 20. So you essentially have 20 different models. And they differ in the way you start your simulation, and the simulation fits it into the constraints that you get out from the big machine. In order to get that model, you still need to stare as an expert at the raw data. Typically this staring at the raw data and refining it takes in the order, so NMR is now completely automated. So for experts in the field, that means that the best experts in the world now need only 1 month to do this. Okay? So this is another difference with respect to X-ray crystallography. X-ray crystallography, most likely everybody in the room could trace, could trace structure relatively soon, or could learn to trace. NMR, you need at least a year to begin even doing one. Yeah?
Is it too optimistic to get only one model with the X-ray and maybe more realistic to get 10 with the NMR and say, okay, I'm not sure, but it could be one of them, because it's something very...
So we enter a debate that I do not like to enter. So NMR, you take the protein in solution, you don't freeze it, so to speak. You determine its structure, you get different solutions because proteins are dynamic objects. This is what NMR spectroscopists say. Crystallographers say, well, in the real cell you have the density of a crystal, so the way we do the crystal, actually what we create is the real physical lowest energy state. So we create the real environment of the protein, we just see it in the crystal, and we take our X-ray beams to look at the crystal, and there is only one solution. And the dynamics, you're right, happens, but it happens around this central solution that I see. And in some cases of my X-ray model, in fact, I see more motion than in others. So I have some idea of motion also in my X-ray structure. And the reason why NMR spectroscopists see many models is because they don't know what is right. They can't make up their mind. So that's the old discussion, the old debate in the field. And you immediately, by your question in some sense, take sides for one of the two sides. So again, the fact is that NMR is much less, and that is because it takes longer. is in this sense more expensive. Just a sec. And the second point that I haven't mentioned yet, NMR is constrained by the length. So NMR cannot do anything much more than 100, 200, 300 residues. While X-ray crystallography can just do anything, in terms of size at least. And that is a big difference, yeah?
Going back to CAST, we need an experimental result which is a perfect answer.
Yes.
But here we...
Yes. So the point is, going back to CASP, he says, for the ones who may not have heard, how can I handle something like this in CASP, where you asked again, do I have different answers? And I kept saying, no, we have one answer. Now he says, but listen, this is not true. I have many answers here, right? So how can I talk about the fiction of a standard of truth if this is the output from my experiment? And the answer to that, in fact, is that CASP uses X-ray structures. Or many people, in fact, but that is really true. So many people have only used one model. But, you know, it's so now we bring it to a different issue. These are problems. On the other hand, they matter only if your prediction accuracy is on the level of that difference. Okay? So if your method is so good that it actually makes a difference whether I take that model or that model, then what you say would matter. But that's not the level of accuracy we reach. So comparative modeling, I said, is the only way to predict reliably structures. And it is not on the level of accuracy of this. Okay? These models in NMR, they agree, certainly if NMR is done well, they agree much more than typically a model and the structure, the template and the target, or target and the template. Let's get back to other methods. And that brings us to the resolution. Resolution is, in some sense, that is cryo-electron microscopy. And again, we have big machines underneath. I don't have a photo of that. Credit card safe. So 4 angstrom, 8 angstrom, 16 angstrom, 32 angstrom. So that is essentially what you see from a large protein. This happens to be GROYEL, which happens to be, you can imagine it like a pot that is used to fold proteins that are not nicely folded. GROYEL is in fact behind one of the things that is behind why I said not all proteins do behave Epstein-like. Anfinsen and Epstein-like. So again, you see much more detail here than on that view. This view, where you see holes in here and can see inside, is the sort of worst resolution for NMR and X-ray crystallography. X-ray crystallography looks into that and NMR as well. So they get structures at 2 angstrom resolution, so way more detail than here. At that resolution, in fact, you know, you see the surface. You see that, in fact, you may see that there are 2 different rings. There's a top part is like a donut. There's a top part of the donut and a lower part of the donut. Not much more. You see some hole in there. But this is the kind of thing you can get with advanced imaging technology with cryo-EM. Now you can use cryo-EM to, in fact, combine cryo-EM with modeling technology and model into what I see. So maybe I just see a blurb, but maybe I can build models for part of that blurb and then fill in the holes. Or say, okay, this level of detail I don't see, but this way of putting my, I believe there are 6 helices, and this way of putting the helices fits with that blurb in just this orientation. So I can combine modeling techniques with a low resolution image that I see and come to a hopefully better image. And that is sort of the thing of the time. Again, back to resolution. So for a particular residue, you see the aromatic ring in very much detail here. That's 1 angstrom, that's sort of 3 angstrom, and models in between. So those are, today's structures are mostly in that sort of level, on that level of accuracy. NMR is more typically on that level of accuracy. Again, NMR sees the motion and has a lot going for it. And here you see that it's difficult to make out really, but the blue net that you may be able to see here underneath, the blue net is what you actually observe. To put the ring in there, you have, in this 3 angstrom, you have different ways of doing it, right? In fact, you find the unique way only if you assume certain rotation, certain distances, and that way you can in fact fit then the molecule into that electron density in blue. While in this particular case here, the density is so high that you can really see the ring. So now you don't need to make many assumptions anymore, or any, if any, because essentially everything fits in here, at least for that tryptophan. Right? Okay, now we can predict structures by comparative modeling. We cannot really do first principle based. Anton is one way, other people try the same, have been trying the same for many years. What we can do, and that relates again to the dynamic solution we see in NMR, we can take a structure that we have, run a computer and see, is that structure that I have really at the energetic minimum? That is something we can do by looking at the first principle or the energy solution. So I can sort of wiggle around. I cannot take a stretched out chain and fold it into the structure. But seeing a structure, starting from an experimental structure, I can say, well, this region is one that is more mobile. And I can do that from a X-ray structure, or I can look at different NMR models. And hopefully, both of those, if they are right, sort of contain the same type of information. And they do, often. But now, let's simplify the problem. Let's just talk about what can we do in terms of really 1D structure. Secondary structure. Secondary structure is stabilized by hydrogen bonds. And I've really taken this to heart and moved out photos of people and CVs of people. But Linus Pauling I wanted to bring in here because of a couple of reasons. One is this set of papers here. I list 5. I do not believe this is the complete number. So we have, when you're a member of the National Academy of Sciences in the US, you have the right to publish 2 papers per year. And so this has changed a little bit, but essentially, this is essentially you can publish whatever you want. Unreviewed in some sense, a peer-reviewed journal. And that is a special privilege that the selected few have. Linus Pauling was one of those. And he published all of these papers, and all of these papers described secondary structure. And different types of secondary structure. Now the remarkable thing is, there are 5 that I quote here, and there are 2 types of secondary structure. How come you can describe 5 papers with 5 types of secondary structure, and only 2 exist? Right, you make mistakes, right? That's simply the case here. Now, the 2 right ones got to Stockholm. The 2 right ones got the Nobel Prize. The other ones are forgotten. Who cares about the wrong predictions if 2 are right? Right? This I find is a remarkable example. So they're just a bunch of these issues where he just keeps going, coming up with things that nobody has ever observed. But then, you know, the 2 right ones are in there too. And you may know that Linus Pauling is one of the few who in fact got 2 Nobel Prizes. The other one for stopping the testing of atomic bombs in the, on the ground. And both of those, unusual also, are unshared. So it's only he. Okay, so here's the idea. Alpha helices and beta strands. Here they're shown the helix. You have hydrogen bonds, so they are stabilized by hydrogen bonds formation between residue I and roughly I plus 4 here. And then we move again, move again, move again. So this is stabilized by these hydrogen bonds that go I and I plus 4. Okay? And that stabilizes the entire helix. Hydrogen bonds are not very strong, but there are many of them. That stabilizes the helix. The beta strand, now we have 2 different parts of the molecule here. That's one part, and that's another part. And again, there are hydrogen bonds every second residue that cross-stabilize. Those are the elements that I talk about. These are the beta strands. And you see that here the interaction comes from somewhere else in space, while in a helix and right here is always I and I plus 4. Again, The beta strand has these hydrogen bonds and it has an orientation. That's what this arrow presentation means. Now in 1954, Linus Pauling got the Nobel Prize for discovering that hydrogen bond formation determines secondary structure, the most important fragment or substructure if you look at a protein structure. When was the first structure done? Some of you may have seen it on the previous slide. I hope not. Any idea? Guess. If you don't have an idea, that's great. That's the situation I'd hoped for. Guess.
1970?
He gets the Nobel Prize in 1954.
Well, that was wasn't that for saying those are the kinds of things that are inside proteins?
Yes. But how can you say that before you see a structure?
Oh, okay.
Was there another answer?
49.
49. It's a great idea. We have a cold winter in 49, so this is a good winter too. So I'm misleading you, of course. The first structure was done in 58, which is in fact after that. And you're totally right, it's theoretical. And then the other one was in 60, Max Perutz, John Kenrue. First John Kenrue and then Max Perutz. Now, there are many remarkable things about this. One is really that Pauling completely speculated before they had ever seen a structure. There was no idea really that structures have helices, or that they in fact would be made up of sub-elements. Now, those were the first two structures we see. What do you believe people believed? Can you see anything? Have you seen enough pictures now to jump at something here? Do you note anything? So the two proteins were myoglobin and hemoglobin. And the reason why they worked on those two is simply because they could get a high density. So those are both X-ray structures. They could get a high density. Somebody asked how the crystallization, you asked how the crystallization is done. So those were simply easy to get the protein material for and aggregated in a high density. Those were the reasons. Myoglobin and hemoglobin. Carrier of iron for oxygen is important for breathing, for the blood. Anyway, so you don't see anything on these two? I've shown you many protein structures. Nothing you stumble over looking at these two images?
There is only alpha helix?
Yeah, that was exactly the conclusion. So that Linus Pauling was really wrong, and even the beta strand doesn't exist. All that we ever have in protein structures are alpha helices. And in fact, that was a prevalent assumption for almost a decade. So I don't know how Linus Pauling felt. But many people, most people seriously believed this is it. How could you define secondary structure from a 3D structure? So I give you the coordinates, but I believe we still have time. I give you the 3D coordinates. How can you get secondary structure out? Oh, we have it sort of, just look at here. So you all see, most likely, that this is a helix by now. How can you, I give you 3D coordinates. I don't give you this blue. I don't give you the shape. I don't give you the cartoon. How can you assign the cartoon? Yeah.
Well, you just do, you just connect atoms that are close to each other locally.
Here?
And you
in this model?
Yes. That's,
you connect so you essentially have a trace between anything that is sort of close to each other in space.
Yes.
And then what? Then you believe it jumps at you?
And then you hope that you can cut out all the false positives and infer something that makes sense.
Let's look at this example here. So the white and the blue. So blue and white are closer to each other, then you begin to see this could be a sort of helix kind of situation here, maybe. but they are closer to each other. And they're not forming the helix. But maybe that would work. Any other idea? So I don't know. Maybe it would work, I don't know.
We could do this method, but only with the carbon backbone.
But that is the carbon backbone. So this in fact is in many ways the simplified view. It has a level of complexity that may not induce in you this idea that this is a simple picture. But this is already only the C alpha, so it's only 1/10 of the atoms.
We can try to make the longest chain possible.
That is somehow, so the idea of the longest sort of chain is proximity. That is a similar idea, right? You label sort of local density in some sense.
No, because we can, if we just look at proximity, sometimes we can find a shortcut
or the network of interaction. But that is, shortcut is for a network, right? I mean, for the contact.
With proximity, we can find a shortcut and say, okay, these two atoms are very close, so they should be linked together, but we can find that if we just keep on going straight and then turn left, we have a longer chain, and this shortcut wasn't effective.
Okay, let me summarize these two ideas. Where you could find you call it a modern image analysis tool that would recognize geometric patterns. And you're clearly right on. This is one way in which secondary structure is assigned by programs from 3D structure. Yeah?
We know what the sequence is, don't we?
Yes.
Yes. So you can look for, so what you need to look in there is for things that are connected and that match the sequence.
Oh, yes, but so again, the goal is essentially to have a program that brings me from that view, or the atom viewer, the atom detail view of that, to something like that. And the sequence does not necessarily help me. Again, it could, and we get back to that on Thursday, but that is not necessarily a sequence problem. And one way that you propose is to find geometric rules, let's call it, geometric ways of labeling it that my eye would be helped in identifying these helices and strands. Any other way?
Yeah, I think I know the answer. There are some ways that the atoms fit together that are allowed, and there are some that are not allowed.
That's a good point. So again, we could have contact potentials or potentials, knowledge-based potentials. He says there are some ways in which atoms can be connected and some ways in which they cannot. And that I can write into a potential, and that potential I can use to assess something. But that doesn't give me secondary structure. Somehow you underestimate the time that I spent on organizing my slides. So the reason why I bring Pauling into this picture is because, you know, that is the great idea, hydrogen bond formations. And in fact, this defined is the idea that you guys have geometry. DSSP is the way to use hydrogen bonds. DSSP, Dictionary of Secondary Structure of Proteins, published in 83, a highly quoted paper. And again, so now what we're doing is first we're going to find hydrogen bonds. How do we define hydrogen bonds? Well, simply we look at the distances between the atoms, and according to the distance we have some threshold. We say if that distance is below some threshold, then I call it a hydrogen bond. Then I label my hydrogen bonds along the chain. And I already said, if I find, so again, according to some energy, I define a threshold. I said that I and I+4 is a helix. So I'm going along the chain, I'm asking, do I see hydrogen bonds between I and I+4 in a continued way? I and I+4, I+1 and I+5, blah, 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 and you build up a helix, right? For a beta sheet, you do it similarly, except now you have two traces that come further apart in sequence, but you still know that the I matches to a J, and depending on the direction, the I+1 to a J plus 1, and so forth and so on. So again, you have a pattern. And then if the sheet is the other way around, you do it the other way around. DSP has essentially 7 states, where H, G, T are helices. There's another one I omitted here. my count is bad, it's eight states. I forgot a pi helix. T is the turnover helix, E and B is beta bulge, so these are sort of a short beta strand. Over here I have a 3-letter alphabet where I have helix, L, E for extended. Extended is really the idea you have something that's flat and long, while the helix is sort of like this, and something loop. Now the way these things are called, there are different names for it. Important thing is a helix, because that is a regular pattern, and a beta strand. Strand, I showed on a later slide, but the difference between a strand and a sheet. A strand is the single thing. You see that is a strand. A sheet is, in this particular case, made up of 4 strands. So much about the lingo. Beta strand, alpha helices are regular, everything else is not. And for everything else, sometimes referred to as loop, sometimes referred to as coil, sometimes as random coil. There are many, many words out there. They're all not really right. The correct one is other. It's just not a helix, not a strand. It's not regular, okay? In that sense, not regular. Now, there are other methods out there. Stride uses essentially expert assignment, fitting to expert assignment. Predictability tries to find secondary structure that sort of is in fact predictable by sequence. That's sort of a very strong idea. Now, once I have these assignments, I can in fact predict secondary structure. And that's what we're going to talk about on Thursday. That will conclude today. Thank you.